welcome to this webinar. And the topic of this webinar is spatial co-expression analysis, large-scale spatial transcriptomics, and we will be focusing on algorithm performance and Stereo-seq data. You might have heard of Stereo-seq. Our sequencing-based spatial transcriptomics technology, Stereo-seq has supported over 100 published research articles, including multiple papers in Science, Nature, and Cell. In addition to transcriptomics solutions, Stereo-seq also offers other solutions like the proteomics product Stereo-seq and the HE-compatible FFPE solution Stereo-seq Omni. Compared to other technology or products, Stereo-seq is compatible with different species. We have evaluated almost 100 tissue types on Stereo-seq. The results have been posted on our official website. Before you start your first Stereo-seq platform, you could check the validated tissue list on our website. First, one of the key features of Stereo-seq is its multiple chip sizes, ranging from less than 1 square centimeters to very large chip size. Besides large chipsets and different species, another strength of Stereo-seq is its single-cell level resolution. These figures represent data from adult mouse brains and mouse embryos, both at cellular resolution. How does Stereo-seq achieve such a high resolution? On our Stereo-seq chips, DNA nanowells are planted at a distance of 500 nanometers, serving as the minimum unit of Stereo-seq data. But in practice, we often combine the spots into larger bins, such as bin 2 means a 2 by 2 spots area, and bin 4 means a 4 by 4 spots area. This concept is important as we will use bin 20, bin 50, and bin 100 data later in spatial co-expression. In addition to combining spots in squares, another method is to use images to separate each cell by their boundaries, called cell bin. Cell bin is more accurate than bin 20 as it combines spots within an entire cell into one data point. Stereo-seq's high resolution and large chip size result in a very large scale data. A single sample may contain hundreds of thousands of bin 20 or cell bin data points, posing significant challenges for data analysis. Now, I will introduce the concept of spatial gene co-expression and next evaluate the performance of our algorithms on our Stereo-seq data. Finally, I will provide specific case studies. You may be familiar with gene-level analysis in bulk RNA-seq, such as WGCNA, weighted correlation network analysis. However, how can we explore spatial gene co-expression network in spatial transcriptomics? Most of the algorithms primarily builds correlations between genes based on gene expression and the spatial location data. The goal is to understand gene interaction networks and identify highly covariant gene sets. We call them gene modules. By adding spatial information, we can better discover disease-related target genes and identify biologically significant functional regions and gene sets. Here, I have summarized 3 application scenarios of spatial gene co-expression from articles. I hope this helps everyone better understand this technology's applications. First, in tumor research, researchers use gene co-expression functions to analyze renal cancer cohorts at different stages. Results show that 14 MYC genes were co-expressed across different stages. These genes can serve as potential prognostic and diagnostic biomarkers for further analysis. Second, in immune research, researchers studied microglia in Alzheimer's disease and found that they exhibit different gene module information in disease and normal states. This discovery allows researchers to recommend some therapeutic targets for the disease state. Finally, in development research, especially when combining spatial information, researchers identify a biologically significant functional region in the mouse cortex, showing as the red circle in the picture. Further analysis revealed that some target genes covary in this functional region, and the subsequent functional analysis indicates that these genes are closely related to social behavior and blood pressure regulation. Spatial gene co-expression algorithms are relatively novel in spatial transcriptomics. Therefore, when searching for relevant literature, we mainly focused on articles from the past 5 years and screen them based on their impact factors and the citation status to assess their suitability for spatial transcriptomic data analysis. In the end, we selected 3 algorithms related to spatial gene co-expression: NEST, hotspot, and HDWGCNA. These algorithms can fully utilize spatial information and are compatible with high-resolution data. Additionally, they do not rely on other clustering and annotation results. They don't affect the integrity of the original expression matrix through data imputation. Therefore, our subsequent algorithm evaluation work will mainly focus on 3 algorithms: NEST, hotspot, and high-dimensional WGCNA. These 3 algorithms has its own advantages and disadvantages. First, the NEST algorithm primarily uses the density-based clustering like DBSCNN to identify the spatial patterns of each gene, thereby further identifying spatially variable genes. Then, after comparing the similarity between genes, it uses graph-based clustering to find co-expression modules. Since it uses the spatial distance information, it can identify high precision or subtle biological functional regions. For example, it can finally distinguish regions of the hippocampus, such as CA1, CA2, CA3, and DG. The schematic diagram clearly illustrates this. Hotspot mainly uses the KNN algorithm to construct a cell similarity graph based on spatial locations. It then compares genes with its cell similarity graph through statistical testing to identify spatially variable genes. Next, it pairwise to compare the similarity between these genes and finally combine them into the spatial co-expression modules through the hierarchical clustering. Its advantages is the use of statistical testing methods, where the corrected p-values are easier to understand, and it calculates gene similarity not only based on the spatial information, but also the phylogenetic information to integrate multiple data types. HDWGCNA was developed by the same team as the commonly used WGCNA, weighted gene co-expression network. This algorithm can be applied to both single-cell data and spatial transcriptomics data. Its principle is to aggregate similar cells into a large cell population, metacells, based on gene expression using KNN, then calculate the weighted adjacency matrix between genes, and finally use WGCNA to construct a gene co-expression network and perform hierarchical clustering to identify co-expressed gene sets. This algorithm demonstrates stronger capabilities in network construction and gene function interpretation. Now, I will share the performance evaluation of these spatial gene co-expression algorithms on our Stereo-seq data. First, let me provide an overview of our test data. We tested a total of 11 samples, including mouse brain, human colorectal cancer, liver cancer, breast cancer, ovarian cancer, lung cancer, and gastric cancer. For each sample, we tested cell bin, bin 20, bin 50, and bin 100 if available. The gene count of bin 20 data ranges from approximately 100 to about 900, indicating a wide coverage of data quality. For the three algorithms, NEST, hotspot, high-dimensional WGC-NA, they can all handle parallel tasks. During the testing, we limited them to use 8 CPUs. For the main parameter settings of these algorithms, we refer to the default parameters in the official tutorials. Only adjusting and adapting the spatial information parameters related to the distance based on our own bin size resolution. The evaluation mainly focused on 4 aspects involving 8 metrics referenced from related algorithm articles and public databases. Since there is no benchmark article for spatial gene co-expression algorithms, these evaluation metrics were selected from the related algorithm publications. We compared the magnitude of results, including the number of spatially variable genes, the number of co-expressed gene modules, and the number of genes in each gene module. We also use Moran index to evaluate the spatial autocorrelation. For the accuracy of spatial co-expression gene sets, we calculate the detection rate of co-expressed gene pairs in public database. And also, we calculate the detection rate of co-expressed gene pairs in public database and evaluate whether genes in the same gene module have consistent biological functions. Additionally, we tested the runtime and the memory usage of each algorithm. Next, I will share the algorithm evaluation results based on 11 test cases using the above evaluation scheme. First, regarding the magnitude of spatial gene co-expression results, we found that hotspot algorithm, the blue boxes in the graph, can detect more spatially variable genes and co-expressed gene modules than the other 2 algorithms. The HD WGC-NA algorithm, the red box, finds more genes in each gene module. Specifically, the high-dimensional WGC-NA algorithm may contain 8 to 12 gene modules, but each gene module may include approximately 300 to 600 genes. Since the number of gene sets and the spatially variable genes found by each sample and algorithm varies, we randomly select the same number of spatially variable genes and co-expression gene set as the baseline, the light-colored box, for a more intuitive comparison of results. Regarding the accuracy of spatial autocorrelation, we found that the NEST algorithm, the green box, significantly outperforms the other 2 algorithms in enriching gene spatial patterns in cell bin and bin 20. As the resolution decreases, possibly due to reduced gene loss, the pattern differs found by the NEST algorithm gradually narrow. In terms of the accuracy of co-expression gene modules detection, we found both gene function consistency and the detection rate of public gene pairs show that the high-dimensional WGC-NA algorithm, the red box, outperforms the other 2 algorithms. Finally, regarding runtime and memory usage, we found that they are primarily related to the number of cells and the genes we use. The more cells and genes, the longer the time required and the greater the memory demand. Overall, the hotspot algorithm, blue box, requires the longest runtime. For Selbin and bin20, due to the large data size, their runtime is significantly longer than bin50 and bin100, exceeding 24 hours. But their memory demand is the smallest among the three algorithms. On the other hand, high-dimensional WGCNA, the red box, and NEST, the green box, algorithms are relatively faster. In the case of Selbin and bin20, the runtime is about 3 to 5 hours, while in the case of bin50 and bin100, the runtime is shortened to 1 to 2 hours. Here is the summary of the algorithm performance testing. The evaluation results of these three algorithms are somewhat correlated with their algorithm principles. For example, the NEST algorithm can better utilize spatial distance information to identify high precision and subtle biological functional regions. In the results, it demonstrates more accurate spatial pattern recognition capabilities, particularly in gene spatial enrichment, and runs faster. Hotspot uses statistical testing method to identify spatial gene co-expression. When we set the corrected p-value to a relatively low threshold, such as 0.05, we can obtain more co-expression modules in cases with a lower number of genes. High-dimensional WGCNA, due to its powerful network construction function, has higher consistency of gene functions within co-expressed gene sets. However, the gene set it identifies may be larger and needs to be adjusted individually based on the specific data. The overall runtime speed of high-dimensional WGCNA is also very fast. It should be noted that due to the limited types and the numbers of samples we tested, the following conclusions are based solely on existing data. Therefore, in specific sample analysis, each of the three algorithms has its own advantages, and you can prioritize the algorithm based on the specific scenario and adjust parameters individually. In the subsequent case studies, I will also mention how to adjust those parameters individually. After we review the performance on Stereo-seq data, let's take a look at some real case studies in scientific articles. First, I will share how to use gene co-expression algorithms to identify tumor subtypes. An article published in Nature Genetics that explores the impact of tumor cell subtypes cell subtypes on the immune microenvironment. The study used single-cell data to confirm cancer cell subpopulation and divided them into MP6 and MP7, two subpopulations. It also identified representative top 30 gene sets in these two subpopulations. Finally, spatial transcriptomic data was used to confirm MP6 and MP7 spatial domain. Based on those gene scoring results, we wonder if we can achieve similar results. Only use the spatial transcriptomic data through our spatial gene co-expression algorithms. Without the assistance of single-cell data, we successfully identified the spatial domain of tumor cell subtypes and find different co-expressed gene modules and their corresponding spatial locations. Here are the results. First, we used the hotspot algorithm and find that we could detect a large number of co-expressed gene modules and identify two gene modules with the largest intersection with the MP6 and MP7 gene sets determined in the article. and we plotted their spatial locations and the partitioning results. The overlap rate of MP6 gene set is 57%, while the overlap rate of MP7 gene set is 33%. At the same time, the partitioning results we find to be quite consistent with the results in the article, with only minor differences showing in the purple region. Hotspots also presents the co-expression modules similarity heatmap to help users to determine the relationship between modules. Additionally, we used the NEST algorithm to achieve the same purpose. We verified its results and find a higher similarity with the regions confirmed by the single-cell data in the article. We can successfully detect the purple region with nearly perfect match. Compared to hotspot, NEST identified fewer co-expression gene modules, but was able to recognize different spatial patterns and classify them into more subdivided regions and layers, consistent with the previous evaluation results. Meanwhile, we also use high-dimensional WGC-NA algorithms to achieve the same purpose. Like NEST and hotspots, we found modules similar to MP6 and MP7, and it can effectively restore the partitioning of tumor subtypes. The high-dimensional WGC-NA algorithm has more sparse scatter spots than the other two algorithms. The advantages of high-dimensional WGC-NA is to further analyze the target genes and map these genes on the UMAP to observe their characteristics. Under the default parameters, these algorithms identify 7 modules, tending to group genes into larger modules. We can also obtain more co-expression gene modules by reducing the parameter called soft power from default 12 to 8 during the network construction. Beyond the common practice of using only mRNA information from spatial transcriptomics, deeper explorations are possible, such as studying the co-expression patterns between lincRNA and mRNA. Through our Stereo-seq Omni product, we can use the NEST spatial co-expression algorithm to identify the co-expression pattern between lincRNA and mRNA in this lung cancer tumor sample. Through research, we discovered a set of MAL81, the lincRNA, and the PTP13, the mRNA, has the same co-expression region located in the tumor cells of this lung cancer sample, which can be mutually validated with cell annotation and clustering results, leading to speculate that these co-expression relationships may be related to the relevant regulatory mechanism. Due to the low expression levels of lincRNA in the sample, the expression matrix is relatively sparse. Using the default parameters would yield fewer results. Therefore, when using the NEST algorithm, I adjusted the relevant parameters such as the hotspot mini size from 30 to 10 and reduced threshold from 0.3 to 0.1 to increase identified co-expression gene modules. Through our Stereo-seq Omni product, we can also further explore the joint analysis of host and microbes. We explore the spatial co-expression genes of the host and the Mycobacterium tuberculosis through a mouse lung model infected after 4 weeks. Using the hotspot algorithm, we observe the co-expressed genes between the Mycobacterium tuberculosis and the host among all the co-expression gene sets we identified, one gene module, including 97 genes, are highly correlated with Mycobacterial tuberculosis genes. We performed KEGG pathway enrichment analysis on these genes and find that they exhibit more active immune activities at certain positions. Notably, macrophage produce a very strong immune response after the Mycobacterium tuberculosis infection, which has also been reported in the related literature. Meanwhile, due to the sparse expression matrix, I attempt to adjust the FDR cutoff to 0.05 and change the model from normal distribution to a Bernoulli distribution to better adapt to the characteristic of the sparse matrix. Finally, we summarize today's presentation. We have detailed why we study spatial gene co-expression and its specific application scenarios. In the performance evaluation, we tested 3 algorithms on Stereo-seq data. These algorithms can utilize spatial information and adapt to high-resolution data. The test results show that each algorithm has its strengths and weakness. We recommend selecting the appropriate algorithm based on specific samples and making personalized parameter adjustments. Generally, if you want to get the results fast, you can try a high-dimensional WGCNA and NEST first. High-dimensional WGCNA shows higher consistency in gene function, and NEST shows more accurate spatial gene recognition. Try hotspot if data matrix is too sparse. Regarding how to interpret and analyze actual samples, we share some practical case studies, including the identification of tumor subtypes and the co-expression of linker RNA and mRNA, also the co-expression of host and the microorganisms. Since spatial gene co-expression algorithms are still a relatively new research field, more algorithms may emerge in the future. We will continue to check these developments and share them with you. The related literature mentioned in this presentation has been listed here. Thank you for listening.